Hi guys, and welcome back to my channel, Biology by Katie. So I recently did a poll over on my Instagram about whether students found longer style YouTube videos or shorter style TikTok videos more useful and effective for revising the A-level biology content. And as you can see, YouTube was a clear winner. So I've decided to allocate more time to producing more YouTube videos over the next few weeks, starting with a topic that many students struggle with, and that is topic 2.4, cell recognition and the immune system. So I really hope you enjoy this video. I hope it makes sense. I hope it clarifies some of those difficult points for you. If it does, if you find it helpful, useful, please do like and subscribe, and you can get to know for any future videos. Let's start this video by looking at the structure of the human immune system. Now, the human immune system is a complex network of cells and organs tasked with protecting the body from any potentially harmful invaders. Our human immune system is split into two major branches, the specific and the non-specific branches. Now, the non-specific branches includes physical and chemical barriers. The skin, for one, acts as a formidable barrier, preventing most pathogens from entering our bodies. Additionally, we have strong hydrochloric acid secreted in the stomach that destroys many harmful microbes that may be ingested with food. Finally, we have tiny hairs in the respiratory tract that may trap dust and microbes and move them away from the lungs. But what happens when pathogens are able to bypass these barriers? That's where the further immune response mechanisms come into play. So we have two types of immune responses. We have the specific and the non-specific immune response. Phagocytes are our body's rapid response team. They engulf and digest pathogens in a process called phagocytosis, which we will explore in more detail shortly. And we then have lymphocytes, which are responsible for recognizing and responding to specific pathogens. So they make up the specific immune response pathway. For the immune system, the ability to distinguish between self and non-self cells is an absolutely key component of the process. So we have two types of cells or particles that may be present in the body. We would have self cells, those that belong to that person, that individual or that organism. And then we have non-self cells, cells that do not belong to that organism and are therefore considered to be invaders. The white blood cells are able to distinguish the difference between cells that do belong to the body and cells that don't, using these specific membrane proteins found on the surfaces of each type of cell. Now, these membrane proteins tend to be glycoprotein and glycolipid structures. So glycoprotein is a carbohydrate chain attached to a membrane-bound protein, and a glycolipid is the same, a carbohydrate chain attached to a phospholipid within the phospholipid bilayer. So the body, specifically the white blood cells, will be familiar with the specific membrane proteins that belong to its own cells, but they will launch an immune response to the membrane proteins that they do not recognize. In this case, we call those membrane proteins antigens, as an antigen is a membrane-bound molecule that initiates an immune response. Let's take a closer look now at phagocytes and the process of phagocytosis. Phagocytes are a type of white blood cell, but they are said to be non-specific. This is because they have a one-size-fits-all response to all pathogens. Phagocytes are a type of white blood cell. They have a nucleus, they have a lysosome, which is a vesicle containing lysozymes, and specifically those lysozymes are a type of hydrolytic enzyme that can destroy pathogens by digestion. The actual process of phagocytosis begins with chemotaxis, where phagocytes are attracted to the site of infection by chemical signals released by damaged tissues, pathogens, and other immune cells. These chemical signals include substances like cytokines. Upon reaching the pathogen, the phagocyte surface receptors then bind to the specific antigens on the pathogen's surface. This binding triggers the next step of phagocytosis, which is a mechanism called endocytosis, where the membrane will engulf around the pathogen, in this case it's a bacteria, to form an intrinsic vesicle that we call a phagosome inside the phagocyte, in which the bacterial cell is now contained. The newly formed phagosome then moves deeper into the phagocyte and fuses with a lysosome, which was the organelle containing those digestive enzymes, the lysozymes. The fusion of these two vesicles, the lysosome with the phagosome, releases those lysozymes into the phagosome where the bacteria is being held. And so as a result, the lysozymes are free to hydrolyze, break down, digest, neutralize that pathogen, in this case the bacteria. So the pathogen is now completely broken down and has been neutralized, is no longer a threat to the body. Once the pathogen has been neutralized, the digested material is actually used by the phagocyte to carry out a process called antigen presentation, which is where the phagocyte will present the antigen of the pathogen on its own membrane, and this helps to stimulate other immune cells. Now let's explore the cell-mediated immune response, which is a crucial aspect of our body's defense mechanism, the specific defense mechanism. T cells, or T lymphocytes, target intracellular disease, such as a cell infected with a virus or a cancerous cell. The cell-mediated immune response begins when a pathogen infects a cell, causing it to display fragments of the pathogen's antigens on its surface. So this could be a body cell that has been infected with a virus. It could be the example that we just looked at previously, where a phagocyte, which is one of the body's own cells, is presenting a non-self antigen on its membrane. Here we have a healthy body cell that has self markers. So these are the unique membrane structures that belong to that cell, and the white blood cells know that. They recognize these self markers. And on the right, we have that same cell type, but this time it has been infected by a virus. So we can see viral particles within that cell. This cell is also presenting viral or non-self antigens on its membrane. So these antigens do not belong to the cell, and therefore the white blood cell will recognize these as foreign or invading. T helper cells, otherwise known as CD4 plus T cells, recognize these non-self antigens and become activated. This recognition, along with additional signals from other molecules, activates these helper T cells, and then once these helper T cells are activated, they begin to secrete cytokines, which are signaling molecules which help to orchestrate the wider immune response. So these cytokines will stimulate the division of these T cells. These T cells will then proliferate into either more T helper cells or cytotoxic T cells, and the cytokines can also go on and activate B cells as well, which we'll talk about in a little while. So as we can see here, we have the body cell that has been infected by the virus in the center. Around it, we have 3 different types of T helper cell, each with a different shape receptor on its cell surface membrane. Only one of these T helper cells is going to be complementary, and therefore only one of these T helper cells is going to become activated. So the 2 T helper cells that have receptors that are non-complementary to this specific type of antigen remain inactive and continue to just circulate in the blood, whereas the complementary T helper cell will become activated and start to divide rapidly so that an immune response can be initiated. So the 3 major roles that these T cells will be grouped into are helper cells, cytotoxic cells, and memory cells. As I mentioned, the T helper cells are there to help coordinate and activate the wider immune response. The cytotoxic T cells, also known as CD8 plus T cells, play a direct role in killing infected cells. They recognize infected cells by binding to the antigen, and upon activation, they release a type of enzyme called perforin. Now, perforin creates pores in the infected cell's membrane, causing it to become permeable, lose its control over what goes in and out of the cell, and of course eventually die. So this induces what we call apoptosis, or programmed cell death. And finally, we have memory T cells, which are another critical component of the cell-mediated response. These cells are long-lived and remain in the body after the initial infection has been cleared. If the same pathogen was to invade the body again, the memory T cells will quickly recognize and they will mount a faster immune response the second time around. And finally, we have the humoral immune response associated with B cells or B lymphocytes. Now, humor, where the term humoral comes from, is an old-fashioned term for blood or tissue fluid, body fluids. So humoral immunity is associated with a response directly to pathogens within the body fluids. So B cells or B lymphocytes target extracellular pathogens such as bacterial cells and toxins. They target pathogens that have not made their way into body cells. So they would target a bacterial cell that is circulating in the blood. In a similar scenario to that of T cells that we have just looked at, there will be many different types of B cells circulating in the blood, each with a different shape receptor on its membrane. If a pathogen does enter the body, it will bind to the B cell with the complementary receptor. Now, once activated, that complementary B cell undergoes a process called clonal expansion, whereby it rapidly divides to produce a large population of identical cells. These cells will then differentiate into one of two main types: plasma cells and memory cells. Plasma cells are the antibody factories of the immune system. They produce and secrete large quantities of antibodies which are specific to the antigen that initially activated the original B cell. These antibodies will circulate in the blood and in the lymphatic system, binding to any complementary antigens which will be on the pathogens, and therefore marking them for destruction by other immune cells. So the antibodies will bring all of the pathogens together in a process called agglutination, which makes it much easier for other immune cells such as phagocytes to come along and kill them. Memory B cells, on the other hand, are the long-term defenders of the humoral immune response. So they do not secrete antibodies in this initial primary infection, but they will remain in the body long after the initial infection has cleared. And if the same antigen is ever encountered again, these memory B cells will quickly differentiate into plasma cells and produce antibodies extremely quickly. So again, we've got a much faster and much more efficient immune response the second time around in a process that we call the secondary immune response. As well as secreting antibodies, B lymphocytes are also able to internalize, which means to take in and present, the antigens of the pathogen to which they are complementary. This then causes them to become antigen-presenting cells, or APCs for short, and subsequently activate the T helper cells. So for full activation, B cells do typically require assistance from these T helper cells. After binding to an antigen, the B cell will internalize it and present fragments of the antigen on its own surface. And what will then happen is helper T cells will recognize this antigen now being presented by a body cell. And so this qualifies for cell-mediated immunity. And so when that T cell binds, that T helper cell binds, both of those cells become active and begin to divide, thus initiating both the humoral response and the cell-mediated response. Okay guys, so because this is a very long and very complicated topic, I have decided to split it into two videos so that it doesn't become too long, take too much time. So that is cell recognition and the immune response part 1 finished. Thank you so much for watching. Hopefully it has provided some clarity to this very, very difficult topic. Please do like and subscribe if you did enjoy the video, and keep an eye out for part 2, which is going to include vaccines, antibody structure, antigen variability, etc., coming very, very soon.